Assalamualaikum and good evening, everyone. Welcome to this webinar session entitled Malaysian Research in Antarctica, Microbes, Diversity, Distribution, and Biotechnological Potential. We are delighted to have everyone here to join us in today's interesting webinar session in conjunction with Hari Kebangsaan 2021 Malaysia Prihatin. I'm Muhammad Faiz Mahmud and I'll be your moderator for today's session. Before we start, allow me to introduce a few basic housekeeping rules. You are welcome to leave comment in the comment box section located on the right-hand side of your screen. If you have any question, kindly ask through the chat box. We'll bring up our speaker who will answer accordingly at the end of the session. We will provide a registration link at the end of our session. So please stay tuned with us until the end. All right, let's start our session now. Before we begin, allow me to briefly introduce our esteemed speakers for today. He is a member of Woosong College and received his PhD in biotechnology from the University of Cambridge in 2000. He was recruited for a university lectureship at the University Malaysia Sabah in January 2004. Prof. Michael, or his full name, Prof. Dr. Clement Michael Wong, is the leader of the Antarctic and Arctic Microbe Research Group in UMS. He has participated in 3 Antarctic and 1 Arctic expedition throughout his life. His group specialized in environmental and molecular biology with a particular emphasis on the diversity of Antarctic bacteria and the bioactive compound they produce. He has extensive scientific link with the scientists at the Universidad de Antofagasta and Instituto Antártico Chileno. It is a privilege for me to welcome our speaker, and we appreciate your effort for taking the time to be part of our today's session. Thank you, Prof. Michael, for joining us today. So without further ado, I would like to invite our speaker, Prof. Michael, to deliver his presentation. So the floor is yours.
Thank you, Mr. Muhammad Faiz, for the kind introductory, and a very good afternoon, everybody. So before I begin, I would really want to thank YPSM for inviting me to give this talk or sharing with all the audience together today. My name is Michael Wong. I'm from the Biotech Research Institute in Malaysia Sabah. So today I'm going to share some of my experiences working on Antarctic microbes for the past 14 years. All right. So I think I was informed that this talk is to encourage more young people, especially school children and also university students, hopefully some lecturers as well, to encourage them to embark on Antarctic research, and hopefully in the near future, some of you be able to participate in Antarctic expedition as well. And I was also made known that the Sota Mizan Antarctic Research Foundation also provides some grants for school children to travel to the Arctic as well. So that is a very good exposure for Malaysia. So just let me begin by sharing my slides. Just give me a moment. Okay. So today I'm going to talk about, as you can see from the title, is a Malaysian's research in Antarctica, microbes, diversity, distribution, and biotechnological potential. It sounds very scientific, but I'll try to make it really light and easy. So I know there are school teachers and some university lecturers as well as university students as well. So I will balance this basically, we'll talk a bit about Antarctic research, my Antarctic research experience. So I'm going to tell you 2 short stories, or rather medium length stories about my pre-expedition winter training in Japan, and also the expeditions that I have been following to the Antarctic continent and also the island. If you can see later, I will talk to you about the research conditions in the Antarctic continent is really quite different from those in Antarctic island, because in terms of cold, it's definitely different. All right. Then we'll talk a bit more focus on about the research collaborations with scientists from Chile for the past 14 years. And that is more on the sharing of experience. I'll show you some of the interesting adventure that we have gone through. And in the second part, we'll touch a bit about science. For those who are really into science, I'll not go deep into it, but just give you an overview what we do and how we carry out the research. And first, we'll look at the microbes diversity and distributions, and then we'll look at a bit about the biotechnological potentials. And towards the end, I'm going to share with you about our major findings and a bit of summary. Where do we go from here? Okay, a bit about the history of the Malaysian Antarctic Research Program. It was established in 1999. That coincidence also, that's the first Malaysian expeditions to the Antarctic. Of course, I was not part of that yet. I joined only much later in 2005, 6 years later from that. And this also marks the beginning of Malaysian Antarctic research. Now, up to today, it has been 22 years. And on record, I think there's more than 100 scientists from Malaysia has been sent to the Antarctic to carry out research. And if you look at it, Malaysia was not a signatory of the 1959 Antarctic Treaty at that time. It means back in 1999. And thus, we cannot launch our own expedition. It means that we cannot go on expeditions on our own. And we rely heavily on collaborations with nations that is already carry out long-term research in the Antarctic. It means that there are 80 members and be able to host us. And we are very fortunate. We have many countries offering us sort of a berth to join the Antarctic expedition under their flags. And before we go to Antarctic, of course, you need to some exposure, especially I'm from the Ecuador area, Khatulistiwa, which is a very hot climate. I have no experience in winter except for some of the experience I have while studying in the UK. So we are lucky to be hosted by the National Institute of Polar Research Japan, or NIPR in short, to carry out the pre-expedition winter training. So give us the exposure before we go to the Antarctic on the survival kits and so on. So we went to Nagano. I think we were taught to do the camping on the field. You can see that the field is full of ice. And Nagano area is previously hosting the Olympic Winter Games. And during that period also, we are also exposed to how to carry out emergency response and how to survive on the ice if we got lost. We are given a compass and a map, how to find a way out of the, like if you get lost in the middle of nowhere. At the same time, you can see that we built tents on the snow, and we have to cook and also sleep on the snow as well. I still remember at that time it was too cold for me to sleep directly on the floor that is snow. But that is the whole idea, is for us to learn how to survive in the Antarctic. You can see some of my Japanese colleagues, and also China colleague, and also Korean that is happily, and here is Anna, one of my colleagues in Malaysia, that we are going through the training. And we also all kinds of if you have been to a scout training camp, you will know how it goes. And then we also walk on the snow, a long journey using the snowshoes. That is quite a bit of challenge for me, especially continuous walking nonstop. But then during the trip, I see a group of elderly, 60 or 70 years old, which walks much faster than me at that time. So I feel a bit bad at that time that I should have walked more. Okay, then leads to after the winter training, then I joined the first expeditions, in the kind invitations from Academia of Science through their collaborations with the National Center for Polar and Ocean Research, NCLR of India. So the expeditions took place in 2004, and it was quite a long expeditions of 3 months staying in the Antarctic. And a lot of talks by YPSM have focused on the work in Antarctic, but I want to share with you really the sea journey. That is very, very interesting to me. And the missions of our first Antarctic trip is to determine the microbes diversity and distributions. And you can see that the icebreaker that we are on for 2 weeks to get to the Antarctic, that is here. And the background, you can see some iceberg there. Okay, just a bit of map to look at how we travel. Of course, we travel from Kuala Lumpur to Cape Town, and from Cape Town, we take the icebreaker. You can see the icebreaker that is moving over there through the ice. And then we go straight down to the Schumacher Oasis, which is the drowning mudland in the east of Antarctic. If you are a geologist, probably you will look at the GPS. It's 70 degrees south. Anything below 60 degrees south is considered the Antarctic already. So we are moving 70. And the journey was really wonderful. It's about 2 weeks, and we've gone through a lot of experience. First, of course, is the very nice water that you see in the tropical, and then, of course, slowly as we move down south, you're gonna see the changes of the landscape. You can first see the ice pancake, because it's called the ice pancake because it really looks like a pancake. But as we go further down, the ice keep on changing. It means that you can see like this ice sheet already, that is on the surface. But as you travel south, the wind becomes stronger, and the ship becomes rolling and pitching. It means that it moves very, very hard, and sometimes the wave can go up to 3 meters high. And that's why there's a saying they call the roaring 40s, which okay, then the furious 50. When you go 50 degrees south, it's really furious, and when you go to the 60 south, it's really screaming. I think those who have gone through the drug passage will experience this as well. So that is a really good experience that you can see. Me and Prof. Papa from UMS share a room cabin in the ship. I can see that all the things falling onto the floor. Some of the even cupboard fall into the floor because of the movement of the ship that is very, very strong. And it's easily, we get a lot of people got seasick. So we are basically most of the time try to endure the waves. And as we go down south furthermore, then the sea will start to calm down already, and you can start to see a lot of packed ice, and you can see penguins running around. That is a wonderful thing. See, I still remember when we see the first iceberg, like the whole ship, the expeditioner of about 60 people, all ran out to see the first iceberg because first time. But after a while, there are so many icebergs around, and people start to get tired of it already. But well, I'm going to share some with you today. And as you go south again, you're going to see a packed ice, very compact ice. And you can see that without the icebreaker, it's very difficult to move beyond this surface of the sea water. And these are the wonderful iceberg that we can see, and it's all around us, and it comes of various sizes and shapes. And these are just some of the real images that you can see as we go around. And sometimes you do find some birds that is floating, penguins, seals. These are quite common along the journey going south. And then eventually you reach a point where the icebreaker cannot move in any further because the ice is just too condensed for the icebreaker to go beyond that. And at that time, the ship slows down, and there's no port for you to dock. You have to rely on stopping in the middle of the sea, and then the expeditioner will have to fly into the continent using helicopters. And you can see very thick ice on the shore. If one of these break away, that you're going to get the sea level to rise. And that's why we are very afraid of the global warming, where a lot of ice is broken away, and then you cause the sea rise, and major cities by the seaside will be affected. And this is the first our trip from the ship to the continent, and you can see the background is the helicopter that we use. And then with me is really Prof. Baba and also Dr. Leila, that is at the time as a student of University of Malaysia. So we flew to the continent. And this is the first impression. You can see that from the top of the helicopter, you can see an ice-free area you call the Schumacher Oasis. And down at the background, you can see a stations belonging to the Indian. You call it Maitri stations. If you look at the whole landscape, you can see that there's no plant or trees. The mosses and lichens are the major inhabitants of this continent, and microalgae as well. And in the Schumacher Oasis, there are more than 100 lakes which are situated in the regions. And during summertime, it melts. It's just a short time. And for your information, Antarctic summer is between sometimes in December and early March. Beyond that, all the lake will be frozen as well. And the beautiful thing, or you call it a bad thing, is that we have 24 hours of daylight. It means that there is no day and night, it's only day on. So 24 hours daylight. And the temperature at that time is about 1 degree Celsius, all right, when we arrive, because it's summer. But by the time 3 months later, when we leave, the temperature already dropped down to -20 to -30 degrees Celsius in our area. I'll show you some of the photographs what happened during our journey. And you can see the two round photographs is actually the mosses and also the lichen. That is most of the organism that you see there. Okay, this is really our first impression from Maitri Station. It's a very big station, that house up to 60 over members at one time during summer. But winter time, there'll be less people just to maintain the station, and those are staying back to carry out some research. And we are happy that the Malaysian flag is raised there, once we are there throughout our journey there. So Maitri is really 70 degrees south and 11 degrees east. And this is the hut that we stay in, that is outside the station. We call it for summer expeditioners, which including me and Prof. Baba. I share a cabin with him inside this insulated cabin here. So it's very warm in there. So the first thing is that we put up our flag there, Jalur Gemilang. And these are some of the other huts by other researchers who have been staying there. And this is a very nice view. Now, this is just behind my tree station. We have a very nice ice shelf. You can see how massive is the ice shelf. At the background, you can see a person there. That is really massive. And we have there's lots of such view around my tree station, which is I would say really a wonderful place to do research. And well, around it, you can see natures, right? And you can see the penguins. This is the Adelaide penguins, which is the one paling tahan lasak. It can stand very, very cold weather. We have other that is milder, which I will show you later, which is the Gen 2 penguins, and also the chin-striped penguins, which cannot withstand the cold that the Adelaide penguins can. It means that this Adelaide penguin is really the one that is tahan lasak. But at the same time, around the Maitri station, you're going to see a lot of very ice formations, like those very sharp one in the ice cave. When you go to the ice cave, you're going to see all this. I still remember all my geography teachers taught me all this, but at the time when I was at school, I don't understand all this and do not appreciate that until I come to Maitri. I can see all this nature. This is really wonderful. And well, you see it's nice scenery there, but those are the good days, man. But during the bad days, you can have snow blizzard like the one I show you, in here, right? You can see some of the expedition, the wind is so strong, it comes with snow that you have to hang on to the rock, otherwise you'll be blown off. So that is how massive the snow blizzard is. It can come very fast and can go very, very fast. So we have to be very careful when we go to the field sampling. Make sure we stay in the group, never go out in a single person. That is very, very dangerous. So we have gone through all this training during the winter training in Japan. So we also have a various briefing on during the sea journey as well. And this is how it looks like, just like how you stay in the fridge. So you can see all the cold environment at that time. But we are provided with proper clothing and things like that, so we are okay. And one thing that you realize that, well, all of us are wearing sunglasses or masks because the UV light in the Antarctic is really higher than normal. So to protect your eyes, it's good to wear the goggle or the sunglasses, all right? So how cold is that? By the time we reached Antarctic, it was quite fine. I'd say it's about 1 degree Celsius. But come to March, we are taking shower and we just put up a pail of water. Then you go back and you realize that it's totally frozen. And when you go to the pea bowl, also all the peas are really frozen there. So that is really an experience. So the water cannot move. I mean, everything gets frozen. Okay, enough about the background. So we talk a bit about science, right? We went for sample collection. So in Antarctic, we don't have cars and things like that. We have some mobile scooters and things like to move around. But if you want to go further, you really rely on the chopper, which we are lucky enough to get the assistance to fly us around to collect sample for research. But some of the nearest research within 6 to 10 kilometers, we have to walk. And these are some of the sites that we have gone to collect some of the samples. We walked and we took some sediments from the so-called lake and also from the snow sample as well. And you can see that I try to work in the lab. Sometimes we prepare all the media, and then the next morning when you wake up, you find that everything is frozen. So that is how the cold is. So to work on the Antarctic could be a challenge as well, unless you have a very, insulated lab where you have a heater in there. All right, so these are some of the photos taken. And basically, we survey this area, the tree marker oasis. You can look at the map down there. All right, this is the Maitri station down here, and then you have another Russian station. It's very isolated. There's only 2 stations in these regions, so very remote. And you probably ask me that, can I have internet access? Now, back in 2005, we don't have direct internet access. All right, we have one by the stations, which we can send out email in bulk one day and to receive email one day, because it's too expensive to have communication like that. But I believe today, 22 years later, is definitely better than back then. See, you can see that we do the sampling throughout this Maitri area. in the west, in the east, and all the lakes we covered. So this area is basically ice-free, and there's 34 kilometers of ice-free area, and it's surrounded by ice. And some of the results that we found that is really interesting, that we found the major bacteria phyla from this region is really the proteobacteria. If you're a scientist, you probably know about this, Bacteroidetes, and then actinobacteria. Then we have also a large population of acidobacteria. But our interest is really on the actinobacteria, Bacteroidetes, and also the proteobacteria. And we went back to the lab and we looked for enzymes that is interesting with biotechnological potentials. And first, we really screened for beta-nanones, which we can use in the textile industry. We can also use in the food industry to carry out degradations at low temperature. So that is the students, Dr. Tam now, who is working on this project. And what we found is that there's one bacteria particularly interesting. We have a lot of bacteria, but one of them is really particularly interesting because express very high manganese activity. So the adaptations of Atrobacter beta-manganese towards the cold temperature involve structural adjustments. You see, it changed. We compared to the manganese produced by tropical bacteria. We found that the structure is slightly different, and that makes it special is that it can active at 20 degrees Celsius as well as 25 degrees Celsius and 20 degrees Celsius. And definitely we have published that in a journal. All right. And that is basically some of the results that we got from our first expedition. Then now I'm going to move on to the a very different experience collaborating with the Instituto Antarctico Chileno. I mean, very nice people. We have been collaborating for the last 14 years, and we have published a lot of papers together. That is since 2007 until now. So this expedition is towards the island. We call it King George Island and South Shetland Islands. That is not the continent. So the temperature is slightly milder because during summer, because it's covered with seas. You see. So the surrounding seawater will actually warm up the continent. I mean, the island a little bit. So a bit of background history and about before our expedition began. Okay, back in 2006, me and also Dr. Azizan, and Prof. Pang Silmo and Dr. Siti Aisha, we all went to a conference in Punta Arenas, the University of Magallanes. At the time, the director, Dr. Jose Ratamales, which is this one here, is very nice, and together with the present director, came to the University of Magallanes to meet the Malaysian counterpart. And soon after the conference, we had a discussion at the INACH office, and where Prof. Dr. Azizan actually presented the Malaysians' interest in Antarctic, what kind of research we are doing, and together with us is some of the INACH scientists. And the collaboration just started there, until today, and still very, very active. All right. And what happened is that after that, then we are invited to join another conference, which is in Conceptions, to meet some of our potential collaborators. We each submitted proposal to INACH, and they try to match us up with some of the collaborators. And in that same year, we flew to meet our collaborators in the University of Conception, Chile, in 2006. That same year, that is where I met with my collaborators, which is this is Professor Gerardo Gonzalez and also Mariana. So these are the collaborators. We have formed a good collaborations, and we have published some papers together. And sure enough, in January 2007, our journey to the Antarctic materialized, and whereby we joined our first expeditions with the INACH. And this one, we don't take the ship. We can opt to take the ship, but we are lucky to be flown in using a commercial flight called a dark flight. And just like the boat, how often you can see that a flight going to the Antarctica, and the flight is at 8 o'clock and it's on time. So that is great. So because sometimes it's not so easy. You plan for a flight and that day the weather turns bad, then you have to delay. Probably if you're lucky, just delay for a few hours. It can delay for days. My second trip actually had to delay for 3 or 4 days because of the bad weather, not in Punta Arenas, but rather in the Antarctic. So we have to delay our flight. Okay, just give you an overview. Okay, this one is the flight that we take. Even that flight is labeled as Antarctica. All right, and this time we traveled to the southmost city of Chile. It's called the Punta Arenas. And you know that Chile is a very long country. You can go from tropical climate up to temperate climate. And from here, we flew to King George Island. I think the journey is not very long. I think it's about 3 hours or so. And it was a smooth sailing journey. We land in King George Island in the Fielder's Peninsula. Of course, in the Antarctic, if you can see, this flight is actually landed in the Antarctic already. And you can see there's no very nice airport like you see around, but it's rather just a flattened piece of land where the flight is allowed to land. And then here is our collaborators. We have Marcelo there, then you have Marianne, and also we have Hirado, Professor Hirado there. And together with Dr. Abirami, at that time was MU students, who is with me traveling from Malaysia to the Antarctic. And these expeditions, our aims is really to determine the microbes diversity of the King George Island area, especially the Fielder's Peninsula, and of course to look for microbes with producing antimicrobial compounds. I will explain a bit later why we are looking for these microbes. And this is the Escudero stations. I think Dr. Alikma also have been to this area, and I believe if she's listening, she will have fond memory of that. Since this is an earlier version of the Escudero, it has not been extended yet. Of course, now Escudero is a much bigger station. I mean, the one in the blue rooftop. And of course, during that day, there's a special day to welcome all the expeditioner. Is also, I think, is a commensurate of their anniversary. You see, so this area is more populated. So you have many stations. You have the Russian station, the China station, you have King Sejong's from Kopri, and also you have the Artigas. That is the Uruguayan stations. You see? Then further a bit out, you have the Argentinian station. So during that day, they invite all these people from different nations to be having a party and to meet each other, especially the scientists at these stations. So you can see the Malaysian flag is one more time flown in the Antarctic. And if you see, like, this place is really wonderful. Compared to Maitri, it's very near to the sea. It's an island, so it's very to the sea. You can see the elephant seals at the background. There are plenty of them. They are basically very not very active anymore, partially because they are on land. I think in the sea, they are very active. You can see plenty of them, all right? But you can only see from far because it can be dangerous if you go near them. And we are not supposed to go near them anyway. And well, these are some of the fur seals that you can see during the expeditions as we travel from island to island. You're going to see some of them lying down. And sometimes they are so camouflaged by the rock, you don't even realize that you're already almost stepping onto it. Because if you look at it, the color is really similar to the rock. And during the expeditions also, when we go sampling, we also see a lot of icebergs. Some of them are really, really beautiful. I can only show some of them, too many photos to show. And this is Atley Island, a protected scientific research area, which we need a permit to get in there to collect samples. So we went into the Atley Islands, and you can see that the Gentoo penguins is really everywhere. It's up to the mountain. And some of the mountain height, I have not been able to show you that the mountain is very, very high. I do not know how the penguins actually climb to the top of the mountain. That is really splendid. And again, these are the Gentoo penguins. They're hatching, the new chicks coming out. Each of them hatching with 2 chicks there. Then there are plenty of them. And then now let's talk about research work. Enough of penguins and also the surrounding. Okay. So basically, we are looking for bacteria exhibiting antimicrobial compound. Partly why we are looking for this compound is that that the discovery of new antibiotics classes has significantly decreased over the last few decades. Means that there are no new classes or very few new classes of antibiotic compared to the rate of developments of multi-drug resistance pathogens. Means that there will be one day, if you don't find new classes of antibiotics, there's no antibiotics to cure even your simplest infection. So that's why the task is to look for new bacteria, new classes of antibiotics in the Antarctic. People have almost exhausted looking all the other continents around the world. Now we have to go to the last continent, hopefully to find something new. That is why we are there. And of course, with all this, we have found some interesting data which have published in the Polish Polar Research as well. And just as we arrive, there's no time to rest. I mean, basically, we have to go down to work, and all right, we travel from island to island with a big ship, and then we have to go down to the Zodiac. We call it small Zodiac here. We have to use that to go to the island to collect samples. So this is one of the sampling site. We have collected samples, and these are some of the results that we found. If you look at here, these are some of the Antarctic bacteria that we found. We test them and see whether they can kill some of the pathogens that's commonly like E. coli, Salmonella, Klebsiella, Enterobacter, Vibrio para, and the gram-positive Bacillus cereus. We found that this Pedobacter BG5 is rather interesting, that it can inhibit E. coli, Salmonella, Klebsiella, Enterobacter, and even the gram-positive bacteria. Whereas the Pseudomonas, a few of others are very potent towards the Vibrio parahemolyticus and also the Bacillus cereus. And so, bingo, actually we have hit the jackpot. Basically, just continue to study them, we might find something new, new antibiotics and so on. And we're also not looking at that, we are also looking at their ability to be resistant or how they can withstand antibiotics. So we can find that the Pedobacter also is resistant to many antibiotics. So that is also a very interesting, and this we have been published in the journal as well. And then one of the bacteria just now that show multiple resistance is that we also found that it carry a plasmid. So the usefulness of plasmid in biotechnological is really enormous. It means that you can create an expression system. You can put in new genes into a bacteria that can survive in the cold. So it's something that is very, very interesting. And this piece of data, we have published it in the Plasmid journal. And then we'll talk about the next expeditions. Now that is in 2008. Right? This time we are really focusing in search for antibiotic-resistant bacteria and also antibiotic-producing bacteria or bioactive compound. And this time we collaborated with Dr. Jenny Blamey, which is is a top scientist in the Chile. He has got her own lab and is very prominent scientist in the Antarctic. And this time I brought along with one of my students, Mr. Tam, and now it's Dr. Tam. And we go sample collection. This time is slightly different. We not only focus on King George Island, but we really go from island to island to collect various samples using the big ship. And then in between, we go down to the Zodiac to get to the island. And this is how the process of moving from island to island. And you can see that we go through very high ice pack, and you see a lot of sea ice floating. All right. And once in a while, we can see whales, seals, and things like that. And this is King George Island I mentioned to you earlier. This is the Fildes Peninsula. All right. The first is that the first expedition, we confine ourselves as the Fildes Peninsula. But this second expeditions, we were allowed or we are lucky to get the collaborations or to provide all the logistics to go around many of the islands, including the volcanic island, Deception Islands, to collect samples. And we have very, very interesting data from Deception Island, which we have published some, and we are still publishing the others. Is at 63 degrees south. Again, we go around, nice scenery. At the same time, we collect a lot of samples, precious samples, the samples that we work until today. And again, Tam, which is Dr. Tam, now we have actually screened a lot of bacteria, up to a few thousands. We found for the 3 strains that is resistant to at least 3 antibiotics, and so on. And definitely we have published those in journals. So if you want to read more about that, you can go to this journal and read about that. And then we have the third expedition in March, but which I did not go myself, but rather is sponsored by Yasan YPSM. One of my students, Jacqueline, all right, he has been to Antarctic and also go around to look for antibiotic producing bacteria. And this time around, we are lucky, we find a very interesting Streptomyces strains that is producing very interesting antimicrobial compound, including those targeting multi-drug resistance bacteria. Pathogens, you see. And what is interesting about this strain is that when the temperature is lower, it has a tendency to produce more antimicrobial compound. And definitely this we have published recently I think it's quite recently in Antarctic Science. There are 2 papers on that. And our main collaborator is really Dr. Paris Levin from INACH at that time, and now is with University Antofagasta. So this is just some of the wildlife in Antarctic island. You can see still once in a while they are fighting every day. So nature is really wonderful, right? So for survival of the fittest fighting is everywhere. All right. And I would just want to summarize this talk here is that what we have, our experience and what we have achieved so far. All right. What we found is that Antarctic really harbors a lot of interesting bacteria with biotechnical potential. I have not talked about the core adaptation strategies. The data that we have is really massive amount of data that we have. But suffice to say that during this short talk here, we have found the Pedobacter species which carry a plasmid, which has the potential to develop into a gene expression system for cold-sensitive genes. And this is really a good biotechnological applications to study more about cold-active enzymes and so on. And the natural bioactive compound produced for survival purpose by this Streptomyces and the Pedobacter and some of the feeder models are really good potential for us to really look deep into the antimicrobial compounds and the antibiotic resistant genes. And we are trying to get grants to continue with this project. And we also find a co-active Atrobacter that have produced enzymes beta-manganese, which has specific use and application, especially in the industry. And we are also working on them as well. And well, this whole expeditions with foreign countries really strengthen our research collaborations between Malaysia and countries with long history of Antarctic research, such as Japan, India, and Chile. Of course, our fellow Malaysian scientists, like the others, have been working with the Australian, the British, New Zealand, Ecuadorian, and many more. And as far as I'm concerned, I'm very happy and also to know a lot of Chilean scientists, which we have been collaborating for 14 years, and it is still ongoing. We have many joint publications. We have 14 joint publications. In total, there are 28 publications just on Antarctic. We are talking about journals only, you see. And we also have mobility between Chilean scientists come to Malaysia, and I've been to Chile as well as my student have attachment at INACH. And of course, we are looking forward to continue this research with our Indian counterpart as well. So that leaves me to acknowledge all our contributors to this project. I would especially like to thank the Ministry of Science, Technology and Innovation, Yayasan Pendidikan, Sultan Nizam, and all the other institutions which have supported this research over the years. I would also like to especially thank Tan Sri Dr. Saleh, and also Professor Dr. Azizan, Dr. Jose Ratamale is the former director of INACH, Dr. Marcelo Lepe, the current director of INACH, and also the Dr. Pandey from NCRR was the former director of NCRR, all right? And all the station leaders and so on, especially UMS has been very supportive over these years. And the people in the team who is on this work is really a long list of people who has contributed to this work. And in fact, this work that I presented is fractions of what we have done so far. Hopefully there's other platform in the future which allow us to share more on this. So these are some of the smiling face has been working behind the scene, working very, very hard to come up with all the results we presented here. All right. And finally, I would like to take this opportunity since our Merdeka Day, and so Selamat Hari Merdeka, then Happy Malaysia Day. All right. So with this, I conclude my presentation, and we'll be happy to answer any questions that you may have. Thank you.
Okay, so thank you, Prof. Michael, for the insightful presentation delivered just now. Personally, I can now see a bigger picture about what we as a Malaysian have achieved. All right, we are now at the end of the first part of the session. So for those of you who are still with us, kindly fill up our attendance from here. And we have already been at our comment section, and you also can scan the QR code there as well. So let's move to the next part of our session. So before that, I would like to remind our audience that you can ask our speakers questions related to our topic today. So feel free to ask in our comment section, and we will highlight it to Prof. Michael. Okay, Prof. So by the meantime, we already prepared some questions for you. So for the first question is, what made you interested to study microbe in the first place?
Well, I mean, if you look at my background, I'm trained in microbiology, especially application in biotechnology. And that's ultimately, made me very curious about these microorganisms. We know microbes, but what happens inside the cell, especially now with the omics era, means that we can really study every single details, every single genes, every single molecule inside the cell. So that is really something very, very interesting already. But on top of that, if you're talking about studying Antarctic microbes, that will make it even more interesting. Like I give you an example, like if you put the microbes, if you put the humans, you freeze them in an ice cube, can they survive? They can't survive. But bacteria, even at -20 and yeast at minus about 12 degrees Celsius, inside the ice cube, they are very, very active. They still carry out active metabolisms. They are not frozen. The surrounding might be frozen, but they can produce some substance like antifreeze protein that makes them still remain active at -degree. So it's really about the curiosity about how these tiny microbes be able to survive in such a cold environment. And you look at a lot of sci-fi movie, you can realize that some of this technology, I'm not saying that it's not possible. In the future, we might preserve a human, for a long time using this antifreeze protein. But of course, now it's still hypothetical that you can only see in the sci-fi movie. But biotechnology, there's endless possibilities that you might be able to apply that in the future. Yeah. So that's the curiosities that really stir my interest. Yeah.
Okay. All right. Thank you, Prof. So, Prof, you have a question from Mr. Ong. There is a saying the water level remains the same when the ice cube melt in the ice water cup. So he wondering why melting of iceberg will be constant, and it is the iceberg could trap more water. And at the same time, Prof, can you explain the difference between the sea ice and the land ice to answer this question?
All right. So I will go for the first one. I'm not a physicist, so I'm not able to answer about the sea level. But I just give you a scenario. You just take a few ice cubes from the fridge, and then you put it on top of a few rocks, you see, in a cup, you see. But once the ice melts, it will definitely becomes increase the so-called the sea level. It's going to increase that. First, you can put it on top of the rock, but once it's melted, it goes to the side, and then you will definitely. If you have a bit of water down there already, the water level is going to rise. This is a bit of a physics question, which I'm not be able to answer directly. That the water level remains the same. That is something that I'm not be able to answer that. But that what I can tell you is that from the IPCC, which is a climate change report, we can say that if the melt of the Antarctic icebergs and all the sea ice and also the large chunk of Antarctic ice, it's going to raise the water level in the oceans. That makes the city that is very near to the sea, just like Venice and things like that, it will be underwater. If it's not, if you don't take climate change seriously. So that is about water level. So the second question is about, is it about the bioprospection or is it something else?
Can you explain the differences between the sea ice and land ice?
All right. The land ice is basically the starting point, where you have a lot of ice there. Perhaps the people studying ice might have answered better. But what my understanding is that when a large chunk of ice break away at the side of the continent, it will fall into the sea and becomes, we call it a sea ice that is floating. So basically, it all started from the land ice that break away from the big continent, from the continent and float into the sea, and it's called a sea ice. Yeah. And on top of the sea ice, sorry, that one is really the iceberg. But sea ice, I think, is a formation that is caused by the cold weather. And that is why you can see that the pack ice, the pancake ice, and so on. I might not be correct, but then I don't have much knowledge about the sea ice. Probably a geologist will be, climatologist will be answer better. Yeah.
Okay. All right, Prof. So next question from Ms. Anna Koma.
Yeah.
Bioprospecting is a tricky topic among polar scientists. So between bioprospecting and the biotechnological potential, how do you see the progress of these studies in the future and your opinions?
Sure. I think it's really the intention. What do you intend to do with it? All right. So when you say biotechnical prospection, there's a gray area between bioprospecting and so on. Bioprospecting is like the end product is more towards commercialization. You want to commercialize something. Biotechnological potential is really not necessarily end up with commercializations. Means that if you find a microbes, you can use it to express gene, then that will expand your knowledge in biotechnology. You can use that microbes, just like I mentioned, then you develop a plasmid for gene expression. Now, once you express the gene in that, it's really more catered towards research to understand more about how, like cold-active enzyme, when you express in the bacteria that is be able to grow at low temperature, then you can understand the kinetics, the dynamics. There's more of understanding. There's no immediate commercial value there. So yes, it's an area. It's really up to scientists what you want to do at the end. If people want to do bioprospecting and make tons of money, that is really bioprospecting. But you can be a scientist doing biotechnological just for understanding or fundamental research. You will just end up there as well and publish a lot of paper, increase our knowledge in that. Thank you. Austin.
Okay. So we have question from Nicole. Where can we read the journal, Prof?
Okay, the journal is normally online, or you can go to the some of the UC library to look for it. If you're not a UC student, then perhaps you can go to Google Scholar, and then you can search some of the journals. Some are open source, which can access immediately, and probably to read every single one of the journals. Yeah.
Okay, Prof. I have a question. So, how the bacteria are able to adapt to extreme freezing temperature in Antarctica?
Yeah, I mean, generally we understand that these Antarctic microbes, they have special mechanisms. I can talk about yeast as well. The yeast, we have a bit more experience in terms of cold adaptation. All right, when everything is frozen, so this is intense. You yourself now, you are frozen on an ice cube, and of course, it's not the surrounding that is frozen, but you inside your body, you get frozen as well. But these microbes have special adaptation. They produce antifreeze protein. So even though the surrounding is actually frozen, but inside the cell, because the ability to produce something like trehalose or something like antifreeze protein, they keep them still in liquid so they can carry out metabolism as well. So it means that we humans, once we are frozen, every part of our body will not work anymore. But microbes, they have special mechanisms. And of course, and if you get frozen, and something very interesting we found in our research is that when it's frozen, you use up all the oxygen. Even though you are fluid, your mitochondria still need to move. You still need to carry out metabolism. You need oxygen, all right? But when you use up the oxygen, what happens? Because it's frozen, there's no new oxygen coming in. They use nitrate as the electron receptor instead of oxygen. That is something that's very interesting that we found from our research. So it means that microbes is multitasking, is be able to adapt to the environment, and you have mechanisms to survive in the cold, at least for a period. Before the ice melt again, probably the next seasons, the oxygen is supplied again. So that is some interesting findings, and we can also look at the antifreeze protein. I think one of my colleagues, Dr. Achira, have found something interesting as well. If you find a gene that is antifreeze, you take it from a bacteria, then from a yeast, and then you put it into a bacteria. And now bacteria has the ability to withstand cold as well. Like E. coli, normally they only grow at 37 degrees Celsius. You put it at lower temperature, they are not happy and they will not be able to grow. But when you put an additional or antifreeze protein or cold shock protein in there, you find that now the E. coli can grow better at low temperature. Yeah. Theoretically, in the future, you might be able to grow something, living things, at a lower temperature by putting some of the genes from the bacteria into this new organism. Yeah.
Okay, Prof. I have another question.
Sure.
So, what is the challenges that you face when working with the International Research Institute, and is it working culture is different from us?
Well, in fact, it's very interesting. I mean, I work when I'm doing my PhD in the UK. It's a different culture. I spent some time studying in Japan as well. It's a different culture. So every country has its culture. There's something good that we can learn from. Definitely something new that we learn is really languages. Like if you go to Japan, you learn some Japanese. Working with the Chilean, you learn some Spanish. About how is the working relationship? I found that most of our collaborators has been very, very friendly and very accommodative. Nice people, I would say, and our research has been smooth going for the past 14 years, no issue at all. All right, and we are happy to welcome them to Malaysia to do the research, and I've also traveled to the INACH to carry out some research there as well. So wow, the collaboration was really, really wonderful. It means nice people. Yeah. And also the Indian has been very, very nice and accommodative as well. I also have a very good working experience with the Indians too. Yeah.
Okay, so Prof, we have a question from Nicole. So can you also talk about the journey studying in uni for this career?
Okay, well, to be frank, when I was studying in the UK and the Japan, I never thought about embarking in the Antarctic. Never in my mind talk about Antarctic. We are really talking about the forefront of research in biotechnology. And you look at back at the time, 2006, biotechnology is, back in 1996 when I was a student, biotechnology is still in the beginning in Malaysia. All right, but in Japan and the UK is already much more advanced than us. That is where I'm really eager to learn more about biotechnology. I still remember my first isolations of DNA in UPM. That's really excites me. That you can really extract the DNA out of there. Although you cannot see it, but then when you run a gel, you can see them. So that really excites me, and I say I want to learn more. I'm from a biology background, and I say that biotechnology is really impressed me, and it can change the world in many aspects, and that's spurred my interest to actually study in this field, working mainly on yeast cells, to look at the fundamental, very fundamental, is looking at chromatin structure. If you know about chromatin, is that the DNA is a long DNA, but it has to pack into chromatin so that it can pack into the small organism so that it doesn't get entangled. So like you and me, we have 23 sets of chromosomes each, or 46 chromosomes in total. Imagine in the tiny cell from your surface of it, if all the DNA 46 chromosome in there is not properly organized into chromatin, if you get entangled, then you'll be in trouble when segregations. So we are really looking into segregations, how DNA move around and things like that. So that's the real interest that's for me until today. But then when I came back and I worked in UMS, it was really interesting story. At that time, we do not know about Antarctic. And then sure enough, you remember our former Prime Minister actually traveled to the Antarctic in 2002. And when he came back, he called for proposal, right? And at that time, I was working in UPM. And I received a letter from our Vice Chancellor to all the faculties, like, who are interested in Antarctic, you have to come up with a proposal the next day. It means 24 hours. Imagine you have no experience working on Antarctic and suddenly you need to write a proposal. So I remember that all night, did not sleep, download a lot of journals and read as many as possible and come up with the proposal and submit it. So I submitted the proposal, I think in 2002, all right? And it went quiet for a few years until 2005, where I received my first call to join the Antarctic expedition. So that is how the bit of story about background, how I got interested in biotechnology and how I get involved in this Antarctic research. Yeah.
Okay, Prof. Question from Rachel. How long was the training in Japan?
The training in Japan is about, I think it's slightly more than 1 week. Yeah. So it is a very intensive one. We have briefing, we have a lecture, and then after that we go for the field. The field expeditions where we have to build camps. We were trained how to save people in case people got like frostbite and things like that. And you don't have the luxury of having ambulance, so you have to make do whatever the surrounding have you that you have. You have to use all this nature to help to save people. And of course, you have to navigate in case you get lost. In the Antarctic, if you get lost, you really cannot see that everywhere is white. There's nothing else. If you have a compass, that really helps you. It's no like Google Map for you to travel around. Because some remote area, there's really no internet coverage, especially the first journey to the Maitri station is very remote. For the second one, where we go to the Antarctic island, especially in King George Island, they have very, very good internet. But that is just within the station compound. The moment you go on board of the ship and touring around the islands, again, you don't have internet signal anymore. So navigations, all this, the training that is provided by, is a very well-run training in Japan. So yes, it's about, I think about 10 days, if I can remember correctly. Yeah.
All right, so Prof, we have another question. So would like to know what are the features of the Antarctica which make it a key venue for microbe research? Are there other places on Earth which offer such features as well?
Yeah, they are. Actually, microbes are everywhere. For instance, if you go to the volcanic area, you're going to find thermophiles. If you go at the bottom of the sea, you're going to find thermophiles as well. And even inside the stomach of a buffalo and cow, you can find anaerobic bacteria that cannot grow with oxygen around. Then why go Antarctic? Don't get us wrong. When we say that we study microbes, we don't just study microbes from Antarctic. We study the microbes from the tropical as well. We have a lot of research going on microbes from Malaysia. But at the same time, you get a chance to find, well, this bacteria that we have in Malaysia, for instance, equatorial area, hot climates, are called mesophiles. Means they grow at higher temperature and they produce enzymes that normally cannot work at low temperature. All right. So to find something that works at low temperature, you have to go to cold countries. Of course, you can go to Japan or Canada to get that. But then those countries they're sovereign countries. You cannot just go in and take sample. Then you are infringing their rights, or you are actually stealing, they call it, from a certain country. But Antarctic is the global continent. If you look at it, Antarctic is a natural devoted to peace and science. In my quote just now, from the Antarctic Treaty Protocol, you can go in and you can take sample. Of course, you go through the ATS system, and you do research on them. And there's no, provided you follow the Antarctic Protocol, then you do not infringe any people's right, because it doesn't belong to any country. And that's why it is a key venue for people to look for extreme. And more importantly, that Antarctic is a pristine area. It means that the area is not disturbed over the millions of years after it's separated from the big continent. It means the bacteria day is really, the original is here. But in Malaysia, for instance, if you go to isolate bacteria from certain parts of it, you cannot be sure whether that's the original place of the bacteria. It could be something that people carry on the boat and step on there, and then you take out the sample, you thought that it's from that area, is actually no, it's from something else. But Antarctic is a pristine that there is a very strict regulation that people cannot just run around, especially the protected area. Then when you go in to just collect sample, you are very sure that it is from that particular place. Yeah, and more importantly, like I say in the beginning, they are cold active. Means the enzyme they produce can be stay very low temperature, and some of the enzymes can work at up to just 4 degrees Celsius. I'm not sure whether this enzyme can work below that temperature, but it will be interesting to find out. Yeah.
Okay, Prof. So as a researcher, how to create interest among younger generation to love science field?
Yeah, I think definitely tak kenal maka tak cinta. So I think that's why we have all this session, is to explain to all our researchers it is possible for you to go to Antarctic. Just like all of you school children, I was a school children as well at that time. When my teacher talked about Antarctic, probably I fall asleep already, because it's not relevant to me at that time. I cannot imagine that Malaysian can go to Antarctic. I didn't even think about I'll be able to go to cold places like the UK and things like that, to study and things like that. So at that time, geographical is really just one of the subjects that I took. And if you want to stir people, you have to let the people know, especially young children now, that it's possible to go to Antarctic. Malaysian already have a very established research group in the Antarctic. So when they grow up, embark on Antarctic, join our groups, and then definitely you get a chance to go to the Antarctic. Yeah, so that is the whole idea. I think YPSM has been doing a very good job. MOSTI has been doing a good job of the roadshow, the outreach program to tell our young scientists that you are be able to go to the Antarctic. Even the person who asked, if you're really interested, I think you can go to the YPSM's website to look for it. I think every year they are called for proposals or competitions for students on ice. Correct me if I'm wrong, Cik Muhammad Faiz, that some of the winners, the chosen one, will be able to go to the Arctic, if not mistaken. Yeah. So yes, in the future, since we are a member of the Antarctic Treaty or acceded to Antarctic Treaty, like I say in the beginning of my talk, is that now we can launch our own expedition to the Antarctic. It's just that hopefully in the future we can have our own station. When we have our own station, then every year we can cater for some school children to participate in the expedition, just like what the other countries doing. Some of them having stations, they always send students during the summer for some experience. Yes, so please go and read up more about Antarctic. You definitely have the opportunity to go there one day. Yeah.
All right. So I guess there is no more question, and we have not enough time. So thank you very much to all participants and to our speaker as well. So with that, we have now come to the end of our session. And before we conclude, allow me to say was a privilege for me to steer this webinar session, which is, in my opinion, was an interesting session delivered by our esteemed speakers. So we hope that it is turned out to be a fruitful session and participants learn something from our session this evening. So once again, I would like to thank you, our speaker, Prof. Michael, from his scholarly and comprehensive sharing, as well as to all participants for cooperation and attendance for today's event. So please stay tuned and like our Facebook page to get updates from time to time for more webinar series in the future, and we will see each other again soon. So Assalamualaikum and goodbye.
Bye-bye. Thank you.
Thank you, Prof.